Okay, so let's talk about something called the electron transport chain. The electron transport chain is the last part of this process that is all included in cellular respiration. So we've set everything up until here: glycolysis, the conversion of pyruvate to acetyl-CoA, and the Tca cycle. This has all used something called substrate level phosphorylation, where a phosphate group from the substrate is being transferred from the substrate to ADP to form ATP. So that's substrate level phosphorylation, and up until this point we have made, for 1 glucose or 1 mole of glucose, we've made 2 ATP in glycolysis and then 6 ATP in the Tca cycle. We've made 8 ATP total, and we know that 1 mole of glucose yields about 32 moles of ATP, or 1 molecule of glucose yields 32 ATP. So we haven't made the majority of the ATP that we can make from the chemical bonds in glucose. So something else aside from the direct making of these ATP molecules that's been happening through here is the loading up of these electron carriers. So NADH, 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 FADH2. We've been loading up these electron carriers. They are reduced, which means they have hydrogens, and they are all going to the electron transport chain. So remember, once we get out of the cytoplasm after glycolysis, now we are in the mitochondrial matrix, which is the inside. So from that, I'm going to draw this. Again, all of this is drawn more clearly on your PowerPoint slides. So absolutely, I would listen to this while you're going through the slides. So we said that the mitochondria has this outer membrane, and then it has this inner membrane, and you'll notice that the inner membrane has all of these switchbacks back and forth. And the reason for this, the inner membrane, why it has these, is called cristae, is because it increases the surface area of the inner membrane. And the inner membrane is where the electron transport chain happens. And the electron transport chain is where oxidative phosphorylation happens. So the more surface area you have on the inner membrane, the more places you have for this energy production to happen. And you've probably heard in biology classes or in some other science classes that the mitochondria is the powerhouse of the cell, meaning this is where we convert all of the energy that we potentially have from different chemicals into this currency molecule ATP. And all of that happens on the inner membrane of the mitochondria. So the reason you see all of these back and forths for this inner membrane is because we're increasing the surface area. Okay, so everything that we talked about, the conversion of pyruvate to acetyl-CoA and the Tca cycle, that all happened in the mitochondrial matrix. And I just wrote mitochondrial matrix. So this is where we converted pyruvate to acetyl-CoA, and then this is where the Tca cycle happens. See why? Okay, so we're right here, and now what's going to happen is we're going to go through the electron transport chain, which are these intermembrane protein complexes that are 3 of them. There's 4 complexes. So 3 are transmembrane. They're transmembrane on the inner membrane. And 1 is peripheral, meaning it doesn't span the membrane, it's just on one side of the membrane. That's complex 2. some pictures don't even show complex 2, they just show complex. So this is complex 1, complex 3, and complex 4. And then the peripheral one is complex 2. A lot of drawings or images in textbooks won't even show complex 2, and that's fine. Some textbooks will also show a 5th complex. But that is really the enzyme ATP synthase. And we said, remember, we're going to make the majority of our ATP from this molecule of glucose in this whole process of cellular respiration. We're going to make it here in this step on the electron transport chain. So what happens here is we're in the mitochondrial matrix. And I'm going to say, so here are these 3 complexes. I'm just going to draw them right here. 1, 2, 3, and then I'll draw this 5th one, the 5th complex here. Again, definitely look at this in the textbook and in the PowerPoint slides, not just on my drawing. So what happens is we have all of these loaded up electron carriers in here, NADH and FADH2. They're loaded with hydrogen. So what happens is the NADH goes in at complex 1. The FADH goes in through complex 2, ultimately through complex 3. The specifics aren't super important, but what happens is these electron carriers are going to be oxidized, meaning that they are going to transfer, they're going to lose their hydrogen ions. And then FADH, this isn't a very good picture, but it will come through complex 3. So essentially what these are is, sometimes you'll see them called proton pumps. Remember, we also call hydrogen ions, sometimes we just call them protons. So what happens is these electron carriers that we now have loaded up with these electrons, these hydrogen ions, throughout all of these processes, once we get to the electron transport chain, they get pumped across, and now we have a high concentration of hydrogen ions in what's called the inner membrane space. So what we're doing is we're creating, it's called the Chemiosmotic Theory. We're creating an electrochemical gradient. And a good analogy for this is kind of like pumping water or having water behind a dam and then allowing the water to flow through, like in a hydropower plant where the water flows through these big turbines. It turns these big turbines and it produces energy. So this is the same thing. Essentially, we pump all these hydrogen ions into this intermembrane space and then we allow it to flow through this ATP synthase enzyme. When it does, it causes a mechanical turn inside this ATP synthase and it forms ATP, this physical movement here of this 5th complex, the ATP synthase. Okay, so this is the point of the electron transport chain. All of these reduced coenzymes, well, yeah, NADH and FADH2 at the electron transport chain, they are going to pump their hydrogen ions. These complexes are essentially proton pumps, increasing the concentration of hydrogen ions in this inner membrane space. And then we're going to allow the hydrogen ions to flow through this ATP synthase, and that's going to cause the phosphorylation of ADP because we have a high concentration of ADP and phosphate already in the matrix. So it's already in here waiting to be phosphorylated. We have ADP and we have inorganic phosphate in the matrix. This enzyme has an active site, this ATP synthase has an active site where it catalyzes ATP synthesis. This is called oxidative phosphorylation because it requires oxygen. And we'll see this in a minute, but what happens is, this is called the electron transport chain, because what happens is when we get to this point where the hydrogens are flowing back across, we have oxygen, which is going to be what's called the terminal electron acceptor. Oxygen is the terminal electron acceptor. That's why this is called oxidative phosphorylation. So oxygen is the final electron acceptor in the mitochondrial matrix. You'll see this, there's a good illustration of this on slide number 64 of your lecture. It shows oxygen accepting the electrons from this reduction reaction and ultimately forming water. It's not just a one-step process, but ultimately oxygen is the terminal electron acceptor and water is the final product. Let's look at the ATP synthase enzyme a little more closely. So the ATP synthase, which some textbooks and some images will call complex 5, there are some good pictures of it. There's a good picture of it on slide 50. There's a good picture of it on slide number 50. It's kind of complicated. I'm not going to do it justice by drawing it, but what happens is there's a transmembrane part of this intermembrane enzyme, and it has a place for each hydrogen ion to come through one at a time. And every time one hydrogen loads in to this enzyme, it causes a conformational change, and it causes this enzyme to rotate one position. So maybe I will draw it. If I draw it like this, so let's say here's the inner membrane, and we'll say this is the inner membrane space where all the hydrogens are. And then, so this is the inner membrane space. I'm just going to write IMS, inner membrane space. So this would be the matrix. So what happens is, and also in the picture it says P side up here and N side. This is really confusing. P stands for positive and N stands for negative. We've created an electrochemical gradient across this membrane. This is the more positive side because of the hydrogens, but this P side and N side, this can be kind of confusing. So embedded in this membrane, this is part of the ATP synthase. This is called the alpha part, and there's 10 subunits. And each one of these subunits can fit one hydrogen in it. Then there's another part. Again, this is way more complicated than what I'm going to draw well, but there's another epsilon part, and then there is a gamma subunit. And then on the matrix side, it kind of looks like a flower with these 6 subunits here. These are alpha and beta subunits, and what happens down here is every time one of these hydrogens comes through, it's going to turn this whole thing one time, and down here waiting in the matrix we have ADP and phosphate, which go into one of the active sites down here in this alpha and beta site, and it produces ATP down here in the matrix. So again, this one, actually this picture was on slide number 49. I've used a lot of different pictures from different textbooks that all show the same thing. Some of the terminology is a little bit different. Some textbooks call this the periplasmic space instead of the intermembrane space, same thing. Some of these pictures are oriented differently because you could have it facing up or facing down depending on which side of the mitochondria it's on. I'm definitely willing or able to talk through any questions you have about this. So this is the electron transport chain. It's made up of 4 protein complexes. 3 are transmembrane that act like proton hydrogen ion pumps, pumping this intermembrane space full of hydrogen ions to create this chemiosmotic gradient. And then these hydrogen ions are going to come through this 5th complex, ATP synthase, one at a time. When it does that, it mechanically turns this rotor and it phosphorylates because each of these subunits has an active site for ADP and phosphate. And when this turns with 1 hydrogen ion coming through, it catalyzes the synthesis of ATP. This is oxidative phosphorylation because oxygen is required and water is a byproduct of this process. Oxygen is the final electron acceptor, water is formed. That's why this is oxidative phosphorylation. One thing that you are definitely going to need to know for the next exam is differences between substrate level phosphorylation and oxidative phosphorylation. All oxidative phosphorylation comes from the electron transport chain. There also is a little bit of disagreement of how many moles of ATP are formed per 1 mole of glucose. But you should be able to, the total yield per glucose is between 30 and 32. We're going to say 32. Well, we'll see. I'll write out what it has in your textbook, but other textbooks show different things. So for glycolysis, let's say how many ATP did we make and how many NADH did we make, or FADH2. Because we make that in the Tca cycle. So in glycolysis, remember we put in 2 and we get back 4, so we net 2 ATP. We also make 2 NADH. The next step was pyruvate oxidation. Pyruvate gets oxidized and the dehydrogenase enzyme converts it to acetyl-CoA. This is 2 NADH. The next thing is the Tca cycle. And because remember here we have 2 pyruvate and we have 2 acetyl-CoA that start the Tca cycle. We have 6 NADH and we have 2 FADH2. We also have 2 ATP. And then we go to the electron transport chain. So let's see what's left. So here we've made, oops. Yeah, I did that right. So we made 2 ATP, so we have 4 ATP, and we have 6, 12 NADH and FADH2. So these 12 NADH or FADH2 are responsible for all the rest of the ATP that we're going to make. I think I made a mistake back here. I'm just realizing when I said that we made 6 ATP per rotation in the Tca cycle, I think I've been saying ATP. This is NADH. We've made 6 NADH and we make 1 ATP, which is a GTP that gets converted to ATP. So I wrote it out correctly over here, but over here on this drawing, I meant 6 NADH and I wrote 6 ATP. So throughout all of this, we total about 32 ATP. So this is the not quite, so these NADH and FADH2 are going to make it's about 2.5 ATP per NADH and about 1.5 ATP per FADH2, but this is not exact. And remember that this glycolysis, these ATP that are made in the cytoplasm. These NADH are in the cytoplasm. They need to get shuttled into the mitochondrial matrix. That takes some ATP. So net, we have about 32 ATP. So I would want you to know where the ATP and the NADH are coming from, the types of cells that this is happening in. Is it aerobic? Is it anaerobic? Is it oxidative phosphorylation? Is it substrate level phosphorylation? And there is a better balance sheet than what I just drew right here. Energy yields from oxidative metabolism. This is on slide number 59. And that shows the net of the whole process. So next we'll talk about regulation of cellular respiration, but glycolysis, pyruvate oxidation, the Tca cycle, and now the electron transport chain, all of these together is cellular respiration, and we will move on to kind of how all of this is regulated more specifically.